Good morning, everyone, and welcome. I'm thrilled to have you here today. Today, we will be exploring the fascinating world of high-performance thin-layer chromatography, mass spectrometry. I'm Dr. S.K. Singh, and I'll be guiding you through this presentation. Over the next few minutes, we'll cover the fundamentals, applications, and recent advancements in this powerful analytical technique. So let's get started and dive into the exciting details. Welcome to our second slide. Today, I want to introduce you to HPTLC, MS, which stands for High-Performance Thin-Layer Chromatography combined with mass spectrometry. It's a powerful hyphenated analytical technique that integrates two core processes: the separation step, which is HPTLC, and the identification step, carried out by mass spectrometry. This combination allows us to not only quickly separate complex mixtures, but also to confidently identify the compounds involved. That's what makes HPTLC, MS especially valuable. It enables rapid screening of samples and provides structural confirmation of the compounds we're analyzing. You'll find this technique widely used across various fields. For example, in herbal drug analysis, it helps ensure the quality and authenticity of herbal products. In the pharmaceutical industry, it supports drug development and quality control. It's also essential in food safety testing and forensic investigations, where accurate identification is crucial. Overall, HPTLC, MS is a versatile and efficient tool that aligns perfectly with the broader theme of advancing analytical capabilities for better decision-making and quality assurance. Let's take a look at the regulatory perspective on herbal analysis. We're seeing increasing acceptance from major organizations like the WHO, which now includes herbal monographs and uses techniques such as HPTLC fingerprinting to ensure quality and authenticity. Similarly, the ICH has developed guidelines like Q2 for validation and Q14 for method development, which are becoming essential for standardizing analytical procedures across the industry. These regulatory frameworks support important aspects like impurity profiling, helping us identify and control unwanted substances, and marker-based standardization, which ensures consistency by focusing on key compounds. However, to meet these expectations, compliance with good manufacturing practices and good laboratory practices, GMP and GLP, is crucial. Additionally, data integrity is a must-have, especially under regulations like 21 CFR Part 11, which emphasizes secure and reliable electronic records. Overall, understanding and aligning with these regulatory requirements not only helps us stay compliant, but also strengthens the credibility and quality of our herbal products in the global market. Let's take a closer look at the principle of HPTLC separation. Essentially, this technique relies on the concepts of adsorption and partition. What that means is that different components in a mixture will migrate at different rates on the plate, leading to their separation. This process is expressed using what's called the RF value, which helps us identify and compare components based on how far they travel. An important aspect of HPTLC is that multiple samples can be analyzed simultaneously, making it a very efficient method. The core idea here is a two-stage process. First, HPTLC separates the components physically on the plate. Then, we use mass spectrometry to identify those compounds by their MZ ratio, which tells us about their molecular weight and structure. What makes this technique particularly powerful is that it provides both a chemical fingerprint, so we know what's present, and the molecular identity, helping us understand exactly what compounds we're dealing with. This combination of separation and identification is what makes HPTLC such a versatile tool in analytical chemistry. Let's take a closer look at the interface principle when coupling HPTLC with MS. The specialized interface plays a crucial role here because it extracts the analyte from the plate and prepares it for mass spectrometry analysis. There are different techniques used for this transfer. One common method is the elution-based interface, where the analyte is washed off the plate using a solvent. In more advanced setups, we also have direct analysis in real time, or DART-MS, which allows for rapid analysis without extensive sample preparation. The key idea is that these interfaces transfer the analyte directly into the MS system, enabling seamless analysis. Now, moving on to the basic principle of MS detection, it involves a few essential steps. First, ionization, where the molecules are converted into charged particles. Then, these ions undergo mass analysis, where their mass-to-charge ratio, or MZ, is determined. Finally, detection records the abundance of each ion, helping us identify the compounds. What this process gives us is a mass spectrum for each compound analyzed. This spectrum is a kind of molecular fingerprint, which we can use to identify and quantify the substances present in our sample. In essence, this interface and detection method are fundamental to obtaining accurate, detailed chemical information from our HPLC plates. Let's take a closer look at the ionization principle. The most common method we use is electrospray ionization, or ESI, which is particularly effective for many samples. There's also APCI, or atmospheric pressure chemical ionization, but it's less frequently used. The key point here is that soft ionization techniques like these are designed to preserve the molecular ions, which is crucial for accurate identification. This approach is especially suitable for analyzing polar compounds and thermolabile substances. They tend to break down at high temperatures, so gentle ionization helps keep them intact. Now, moving on to the elution and transfer mechanism. Typically, we select a band, which can either be scraped off or eluted, and then the solvent extracts the analyte from that band. This step is vital because it concentrates the analyte and prepares it for detection. Once the analyte is extracted, it's introduced into the mass spectrometer primarily through electrospray ionization. This method allows us to directly identify the separated spots from the chromatography, linking the separation process with mass analysis seamlessly. The basic principle behind ESI is that it produces charged droplets from the solution, which then evaporate, leaving behind charged analyte ions. This process is gentle and effective, enabling us to analyze complex mixtures without fragmenting the molecules. So, in summary, the ionization process, particularly ESI, is central to many analytical workflows because it maintains the molecular integrity of polar and thermolabile compounds, allowing for accurate, direct identification in mass spectrometry. Now, let's take a closer look at the mass analyzer principle. Essentially, this part of the mass spectrometer is responsible for separating ions based on their mass-to-charge ratio, or M. This separation is crucial because it allows us to identify and quantify different ions in our sample. There are several common types of analyzers, and three of the most widely used are the quadrupole, the ion trap, and the time of flight, or TOF, analyzer. Each of these has its unique way of filtering ions, but they all aim to achieve precise separation based on MZ. What makes this process so valuable is that it provides us with accurate mass determination. This accuracy is key in identifying compounds, especially when working with complex mixtures. So, in the context of our overall theme, understanding these analyzers helps us appreciate how mass spectrometry delivers detailed, reliable information about our molecules of interest. On this slide, we're looking at the two main outputs from our detection process. First, we have the HPLC chromatogram, which is based on the RF value. This gives us a visual representation of how compounds separate on the plate. Then, we also get the mass spectrum, which is based on the MZ, or mass-to-charge ratio, and helps us identify compounds at a molecular level. What's really important here is that these outputs enable dual confirmation. That means we can verify the compound's identity in two ways: by checking its retention, or RF value, and by confirming its molecular identity through the mass spectrum. This dual approach significantly increases our confidence in the results, ensuring they're accurate and reliable. Connecting this to the overall theme, these detection outputs are crucial because they provide both an initial separation profile and a detailed molecular fingerprint. Together, they give us a comprehensive way to confirm what compounds we're dealing with, which is essential for precise and trustworthy analysis. Let's take a closer look at the quantification principle. This approach is based on multiple techniques, including densitometry, specifically HPTLC, and MS signal intensity. By combining these methods, we can significantly improve both accuracy and sensitivity in our analysis. This integrated approach is particularly powerful because it supports multi-component analysis, meaning we can quantify several substances simultaneously with greater confidence. Overall, this synergy enhances our ability to obtain reliable, precise results, which is crucial for complex sample analysis and ensuring the robustness of our analytical methods. Now, let's turn our attention to system suitability and compliance, which are fundamental to ensuring our analytical methods are both reliable and compliant with regulatory standards. System suitability encompasses several critical aspects, starting with RF reproducibility, which ensures that our chromatographic separations are consistent across runs. Then there's MS calibration and mass accuracy, vital for precise identification and quantification of compounds. On the compliance front, we're required to adhere to regulations like 21 CFR Part 11, which governs electronic records and signatures, ensuring data integrity. SOPs and audit trails are also essential. They provide documented procedures and traceability of all actions taken during analysis. Controlled documentation further supports this, making sure all records are accurate, complete, and compliant. Method validation, guided by ICH-Q2, is another cornerstone. It involves verifying key parameters such as specificity, linearity, accuracy, precision, as well as determining the limits of detection and quantification, LOD and LOQ, and robustness. These parameters are not just technical checkpoints, they're critical for gaining regulatory acceptance. By rigorously addressing system suitability and compliance, we uphold the integrity of our data and ensure our methods meet the stringent standards required for regulatory approval and reliable scientific results. Let's take a look at some of the key advantages of this method. First, it uniquely combines separation and identification in a single process, which really streamlines the workflow and enhances accuracy. This makes it particularly ideal for herbal fingerprinting, where identifying complex mixtures accurately is crucial. Another big plus is the minimal sample preparation required, saving us time and reducing potential errors. And when we compare it to traditional LC, MS techniques, it's definitely more cost-effective, making it accessible for more labs and applications. Overall, these advantages really highlight how this approach can improve efficiency and reliability in herbal analysis. For further support, please don't hesitate to reach out. We're here to help you with any questions or additional information you might need. Feel free to contact us through the provided channels. And with that, I want to say thank you for your time and attention today. I appreciate your engagement and hope you found this presentation useful. Remember, we're here to support your success moving forward. Thank you again.